Hello, in this video we're going to talk about hemostasis, or the maintenance of blood within blood vessels, right? So to appreciate hemostasis, one should understand the structure and the function of platelets. Why is that so? Platelets are the major characters in hemostasis. Hemostasis is also the prevention of blood loss through the formation of a blood clot. So platelets are the ones which form this clot. They are also known as thrombocytes, and they are minute discs which are 1 to 4 micrometers in diameter. They are formed in the bone marrow from megakaryocytes. So this is what happens. We have a multipotent hematopoietic stem cell which is activated via its receptor by transcriptional factors. It then differentiates into a common myeloid progenitor, which again is activated by thrombopoietin, and it divides into a megakaryocyte and forms platelets. Yes. These platelets are actually fragments of megakaryocytes. They are formed when platelets squeeze through capillaries, right? And the normal concentration in blood is between 150,000 to 450,000 platelets per microliter. And 1/3 of these platelets is trapped in the spleen. So people who have a condition such as thrombocytopenia will require a splenectomy, that is removal of the spleen, so as to bring about normal concentration of platelets in blood. Platelets are very important. They have a lifespan of 7 to 10 days, and they have functional characteristics of whole cells, even though they do not have nuclei and can't reproduce. So we see that these platelets have a coat of glycoproteins on their membranes, which repulse adherence to normal endothelium but cause adherence to injured areas of vessel walls. This is so as to prevent unnecessary clotting, which is very dangerous, as it will lead to thrombosis, and also it can lead to high blood pressure, which is not good for you, right? We also see that it has phospholipids, the membranes, which activate multiple stages in blood clotting, as they have embedded within them membrane receptors for agonists. So these agonists, such as collagen and von Willebrand factors, activate these platelets. So the first one here, we have glycoprotein 1A, which works on collagen when there is blood exposure to collagen due to injury. Then we have glycoprotein 1B, which works on von Willebrand factors, which are found on the endothelium or on the subendothelium, which adhere and activate them. We also have glycoprotein 2B3A complex, which again works on von Willebrand factors, and also fibrinogen, which I'll show you in the upcoming slides, right? Now, the cytoplasm of these platelets, it contains contractile proteins, it contains mitochondria, it contains remnants of Golgi apparatus, and it contains granules. Of these granules, we have alpha granules, which are secreted proteins such as von Willebrand factor, fibrinogen, and factor 5. Then again, on granules, we have dense granules, which are non-protein granules, which contain non-protein substances, such as ADP, which attracts more platelets to the site of injury, ATP, which then is broken down to ADP, then serotonin, which is a vasoconstrictor, and take note, vasoconstriction is the first response to blood exposure to collagen, or first response to vessel damage, right? Then also calcium. Okay. Then within this platelets, we have a very important growth factor, um, which is called platelet-derived growth factor, or PDGF, which is a mitogen which has two subunits. This mitogen works on its receptor called PDGFR, or platelet-derived growth factor receptor, and this complex brings about migration, proliferation, differentiation, and survival of many of these cells, such as fibroblasts, smooth muscle cells, endothelial cells, and so on. Right? We also see that this complex is important in the formation, or in the coding for the formation of new blood vessels from pre-existing ones. And you also see that in platelets, when they are released, right, they kind of attract even more platelets to the site of injury, right? So they are very important. And since they cause proliferation of cells, they can be used in the pathology of cancer, right? So they are used by scientists in cancer research. So scientists are trying to produce antagonists against these complexes, so as to prevent the spread of cancer, right? So what then regulates platelet production? We have stimulating factors, colony stimulating factors, and thrombopoietin. And like thrombopoietin, like erythropoietin, thrombopoietin is produced by the liver and kidneys. And this thrombopoietin, it activates platelets, it activates the lineage in itself, the thrombocyte lineage. So this thrombopoietin is actually very important, right? It works on the receptor of platelets. So it leads us to this point. When platelet concentration is low, unbound thrombopoietin will be high. And when platelet concentration is high, unbound thrombopoietin will be low. Take a minute or two to meditate upon this. It's very simple, right? Now, thrombopoietin, it increases megakaryocyte production and growth, and it stimulates growth of the erythroid progenitor cells. That is this progenitor cell and this progenitor cell. It stimulates them, and it stimulates proliferation and prolongs the survival of the hematopoietic stem cell, right? By stimulating, leading to its proliferation, right? In addition, it makes the platelets more sensitive to agonists, which we said are Van Brand factor and collagen, right? It makes them more sensitive to them, and it will lead to their aggregation or recruitment to the site of damaged blood vessel, right? Now, hemostasis, right? As we said, is the formation of blood clots in walls of damaged blood vessels to prevent blood loss and maintain blood within the vascular system. So there are four basic stages. The initial response being constriction of blood vessels and formation of a temporal platelet plug, and the secondary response being activation of coagulation cascade and formation of a plate of fibrin plug, right? That is, primary hemostasis is initial response, secondary hemostasis is the secondary response, right, which is dependent upon clotting factors, and it will lead to activation of coagulation cascade and formation of a fibrin plug. Now, this is the formation of a temporary platelet plug in a nutshell. Right. So in the primary hemostasis, firstly we constrict our blood vessels to prevent blood loss. Then we form a platelet temporal plug, right? So these platelets, they adhere to Van factors on the endothelium and collagen, and this will lead to the shape change of these platelets, right? And this will then lead to their degranulation or release of granules such as thromboxane A2, ADP, serotonin, and these granules, they lead to, or they are cytokines, they are chemical messengers. They will lead to the recruitment of more of these platelets, right? So another diagram to help you understand again, we have glycoproteins on the surface of these platelets. Due to injury, they are exposed to collagen and they bind to collagen. This will lead to their shape change. As you can see, they were once circles, now they are kind of hexagonal, and it will lead to their degranulation. Can you see the granules were first contained within the platelet, now they are moving out. A more clearer diagram here, they bind here to these collagen and Van factors. They release granules ADP, PAF, these platelet factors, and thromboxane A2, right? Which will lead to the recruitment of more and more of these platelets to the damaged blood vessel, right? And to impaired blood flow. It's a temporary platelet plug. Now, how then does this platelet plug form? We see that the platelet plug forms when there is fibrinogen between two of these platelets, right? So fibrinogen will be meshed between two of these platelets. The platelet plug is as of a result of fibrinogen being connecting the two platelets, right? That is the essence of platelet plug. Sorry, platelet aggregation, right? So this again, another summary to help you remember and understand. We have injury to our blood vessel wall, and in the blood we know that blood contains platelets. So these platelets then move and run towards this collagen and Van factors, and they attach. And when they attach, they change shape and release granules. They degranulate, and these granules, they then attract more and more of these platelets to the site of injury, since they are chemical messengers, and they start to form a plug and aggregate. They aggregate when between fibrinogen via the fibrinogen receptors, right? You see, they are attaching and they are impeding blood flow, right? So that's all for now. If this video helped you, make sure to subscribe. In the next video, we're going to talk about secondary hemostasis. Yeah, so thank you for watching. See you in the next video.